Welcome to Chapter 3 of Human Biology. This chapter focuses on the cell structure and its function. So what are the parts of the cell and what do they do? At the end of this lecture, we should be able to answer these questions. What is cell theory? What is the difference between a prokaryote and a eukaryote? And can you describe the parts and functions of eukaryotic cells? That is the type of cell that we have, so that's what we're going to focus on this semester. How do things travel into and out of cells? So how do they go across the membranes of our cells and travel around inside the cells to get where they're going? How does osmosis affect cells, the movement of water? And what is cellular respiration? Last time, in the last chapter, we ended with kind of an overview of the major molecules that make up our cells. In this chapter, we'll see how those molecules function and interact with one another to create the structure of cells, influence their activities or the type of cell that they are, and form energy for use by the body. So as a quick review here, our major classes or types of molecules are going to include our carbohydrates, our sugars, and our starches. These provide our quick energy sources for the body. And this image is of a complex carbohydrate like glycogen, and we can break those bonds, break them off to get these individual monomers of glucose to make energy out of. We have our lipids. Here's a triglyceride with our glycerol head and our fatty acid chains or tails, and our all-important phospholipid. This is going to be the star of our focus today. The phospholipid having the glycerol head with a phosphate group there and two fatty acid tails. And these guys we have our hydrophilic heads and our hydrophobic tails, and that interaction and how it interacts with water is going to give us the structure of our plasma membrane. Just a little preview. And then lastly, we have our proteins, our polymers of amino acids. The proteins are the molecules that are performing all of the work within the body. So our DNA tells the body what protein to make, but the protein is the thing that goes out and completes the task, makes us look the way we do, gives all of our enzymes. Remember, enzymes are examples of proteins moving. So we're going to need all of those in order to understand what is going on inside of our cells. First, let's start with cell theory, and of why we focus so heavily on cells. And the cell theory is one of the fundamental principles of biology, and it really influences how we think about and study living things. And there are three main tenets here of the cell theory. First, cells are the smallest unit of life. So we talked in the last chapter about the atom being the smallest unit of matter, but all things, whether they're living or non-living, are made of atoms. If something is living, biotic, then it has to be made of one or more cells. Second, cells make up all living things. And I've always thought of these first two as pretty much saying the same thing, but we leave it to scientists to be as absolutely clear as possible. So cells make up all living things, and the smallest unit where they can be considered something that's alive is the cell. And lastly, new cells arise only from pre-existing cells. So existing cells will replicate themselves, whether it be mitosis, making an exact replica, or meiosis, where we're recombining some of our DNA. But those cells have to get that information from a pre-existing cell, one that already exists. Let's do a quick review here of our domains of life. So we are in domain eukarya, with the plants and the fungus and all the other animals. We have eukaryotic cells. That's not the only type of cell that exists. We also have these two other domains, bacteria and RK or Archaebacteria. And these two domains are made up of what we call prokaryotes or prokaryotic cells. So let's look at some of the differences between those two. Prokaryotes tend to be smaller and more simple in structure than the eukaryotic cells that we're going to talk about next. A prokaryotic cell does have a plasma membrane around the outside, but it does not have any internal membrane-bound organelles, and that includes the nucleus. So the DNA of a prokaryotic cell clumps together in a blob, but it's still kind of floating around in the nucleus. There's nothing surrounding it to protect that. In this image of the E. coli bacteria, this is a prokaryotic organism that we've probably all heard about. And this black blob here in the middle, there it is, the black blob here in the middle is the DNA, not bound by a membrane. So this is a prokaryotic cell. Interestingly, prokaryotic cells do have ribosomes. So that is going to be an organelle that we will see in a eukaryote as well that also exists in our prokaryote. Domain eukarya, where we are, includes plant cells, animal cells, and fungal cells, and the biggest difference is that they have membrane-bound organelles. There's a lot more diversity in shape, structure, and function among these eukaryotic cells than we see in prokaryotes, and a lot more diversity in the forms of life, as you can see. If you're not familiar with the term organelle, that means that it's a tiny organ. So just like we have organs that perform specific functions, our cells have little pieces inside that perform specific functions within each cell. And we're going to go through those all here in just a second. So we saw that our prokaryotes have plasma membranes around them, and so do our eukaryotes. And there's a lot more diversity in how that can look in eukaryotic cells. So here's our typical animal cell, and we're going to look in a lot more detail about the plasma membrane of that. But then we have our plant cell that has that rigid cell wall reinforced with cellulose to make it very strong and rigid and keep that plant standing upright. Then we have our fungal cell, and fungal cells just always blow my mind because you can kind of tell by looking at it that it's really more closely, has a lot more similarities to an animal cell than a plant cell, even though their lifestyle and the way that they go about survival is more related to plants. When discussing any kind of biology, we often hear the phrase form or shape follows function. And this just means that the structure of a cell or body part can tell us a lot about its function in keeping the organism alive. One example that we'll talk about a lot in this lecture and throughout the semester is red blood cells. So red blood cells are not round like a typical animal cell, but they're kind of a biconcave disc, or almost like a donut, but not hollow in the middle. And this unique shape and the fact that they're pretty small compared to other cells gives them lots of surface area to carry out their primary function, which is to carry oxygen through the bloodstream to cells and carry carbon dioxide away from cells. Very important task. If they were round or larger, like the typical animal cell, then we would need a lot more red blood cells to accomplish the same amount of work. So making these cells more specialized and efficient also decreases the amount of energy that we have to spend maintaining and replicating those cells. So the fact that that cell has a specific shape that lends itself to that function is that idea of form follows function. So let's look at the plasma membrane before we jump into our individual organelles. And as we've already seen, the plasma membrane is made up of a phospholipid bilayer, so 2 layers of those phospholipid molecules. And they have embedded proteins and carbohydrates. Proteins are embedded in the bilayer or attached to the surface on either side of the membrane. So whether it be the outside facing the extracellular space or the inside facing the cytoplasm. Carbohydrates, mostly cholesterol, attach only to the outer surface of the membrane. And these are very important for keeping the membrane flexible and able to be movable and not rigid. We describe the plasma membrane as being what we call the fluid mosaic model. And the proteins, the carbohydrates, and the lipids, those are like the different colored tiles of the mosaic, but we call it fluid because many of those embedded proteins are really able to move laterally or side to side or around in that structure of the plasma membrane. So they're not completely stationary. One of the key characteristics of a cell's plasma membrane is that it's semi-permeable. And I really like to think of it more as being selectively permeable, meaning that it will allow certain molecules through, but not all. And it's being selective because the types of proteins and carbohydrates embedded in the membrane are going to dictate what substances it will allow into and out of the cell. And these are going to change from cell to cell depending on the type that it is. So the plasma membrane is kind of like the gatekeeper or the bouncer, making sure that only things that are helpful to the cell or not harmful are going to be allowed into the cell. What forms this bilayer structure? That is really the interaction between the phospholipid molecule and the water. So there's water surrounding the cell on the outside, and there's water that makes up a big portion of the cytoplasm on the inside. And our hydrophilic or water-loving heads are going to orient themselves towards the inside and the outside because they're attracted to that water, that polar molecule. The hydrophobic tails are going to be oriented towards the inside of the cell, repelled by that polar water molecule. So that is what gives us this phospholipid bilayer shape. And then the fact that it's able to embed all of these proteins and carbohydrates in there is what is going to give it its function. So what are the functions of the plasma membrane? First, we've seen that it helps keep its structure. This is what surrounds the entire cell. It's going to help keep the extracellular fluid outside and the cytoplasm inside, and regulate the movement of substances into and out of the cell. And we want to remember that it's called selectively permeable because it will allow some things to travel in and not others. And that's going to be dependent on the type of molecule. Can it interact with lipids? If it can, and it can diffuse right through the plasma membrane, then it's allowed in. If it can't, and if there's no channel that will let it in, then it's not going to be allowed in. The plasma membrane also functions in cell recognition. So we've got glycoproteins here, and these act kind of like name tags, allowing cells to identify if they're known or foreign. And this is how our bodies recognize things like bacteria. If it doesn't have the same glycoprotein as the organism that it is currently in, then it must be a foreign thing. And we can tag that and mark it by our immune system to get rid of it. The plasma membrane functions in communication between cells through the use of receptor proteins embedded in that bilayer. Not all cells have the same receptors, so they're capable of receiving different messages. We're going to see a big example of that when we talk about the endocrine system and how hormones send their messages through the bloodstream. And lastly, the plasma membrane allows cells to bind together through the use of cell adhesion molecules. So again, another protein that is embedded in the plasma membrane, and this is what allows specialized cells to group together and form tissues, which can then fuse with other tissues to form complete organs. So those cell adhesion molecules, those CAMs, are what's keeping our individual tissues together, which we will see are made up of a bunch of different cells. Okay, in part 2, we'll talk about how things move across this plasma membrane. See you there.